Bats are not the easiest creatures to study. Most of the world's 1,200 or so species lurk in the shadows, emerging only at night to feed. So you have to stray from the beaten path to enter their world, and you have to seek them out on their schedule, especially if you want to study the myriad pathogens that infect the animals. Biologist Paul Cryan has been here before. It's a remote study site in the Rocky Mountains west of Fort Collins, Colorado.
This is one of the sites where we sampled bats for a coronavirus study several years ago, and we did have some samples that we detected new coronaviruses that had never been described before.
Cryan and his colleagues haven't found really nasty viruses at this site, but bats elsewhere are known to carry some of the most dangerous pathogens around: SARS, MERS, and Ebola, to name a few. And yet little's known about how they harbor and transmit these diseases. This vacuum of information draws scientists like Cryan to brave lightning storms to collect data.
So let's give it a few minutes, see what happens with this, and if it's still looking sketchy, we'll head down the hill.
They were hoping to net a variety of bat species that visit a small alpine pond nightly to drink. The idea was to sample the bacteria on the surface of the bats to characterize the animal's skin microbiome. Heading up that project is Christine Avina, a PhD student at the University of Colorado Boulder. She's gathering baseline data to study how the microbiome of bat skin might interact with white nose syndrome, caused by a fungus that has devastated bat populations across the U.S. in the past few years.
The next step would be moving into either how that environmental bacteria can get onto the bat and what the different caves might have in them, the white nose caves versus the non-white nose infected caves, and also just the white nose individuals versus the non-white nose individuals.
But alas, fieldwork is nothing if not unpredictable, and the weather finally forces the team off the mountain. But science marches on, and back in town, researchers at the Centers for Disease Control and Prevention's Division of Vector-Borne Diseases are uncovering other bat secrets in the comfortable confines of the lab. Cliff McKee studies the bacterial genus Bartonella, which can cause human diseases, including cat scratch disease and trench fever. It's also carried by several bat species, including the large African fruit bats that McKee studies.
We have blood samples that have been taken from Eidolon helvum and Eidolon dupranum, which are the straw-colored fruit bat and the Madagascan fruit bat in Africa. The traditional way we would characterize an infection in a reservoir is through culturing, because that's kind of the smoking gun for an infection. The primary work I do is working directly with blood. I'll take the blood samples and extract DNA from them, and I can do genetic characterization directly from blood.
McKee has developed a multi-gene PCR platform he uses to isolate sequences from 6 different genes across the Bartonella genome. This technique can detect all the bacteria hiding in a given blood sample.
From DNA to sequences, target positive Bartonella sequences, I can get within a day.
McKee and his CDC colleagues have so far found 6 different Bartonella species in the bats they study. Many bats can even be co-infected with several species of the bacteria simultaneously.
We see very high prevalence, upwards of 60–70% of bats are infected.
And even though bats don't tend to show symptoms from Bartonella infections, they could be passing on the bacteria to other mammals, including humans.
2012, a human case was reported in Wisconsin. A person showed symptoms of endocarditis, and they isolated a new Bartonella species, Bartonella myotiminensis, from his heart tissue.
Later, scientists found this same species in a bat, representing the first documented connection between a human Bartonella infection and a bat host.
We haven't seen any human cases of Bartonella that have been connected to Eidolon bats. However, there are multiple risk pathways that could be, allowing spillover to occur.
In addition to monitoring the spillover potential, McKee and other researchers are learning how bacteria and viruses evolve in the veritable hothouse of the bat immune system.
We've seen that Bartonella shows this, high level of flux in its genome, where it can exchange genetic components at will with other co-infecting Bartonella species.
Processes like co-infection, recombination, and lateral gene transfer can accelerate the rate at which bacterial evolution occurs in bats.
So using the system, we can start to understand some of those direct genetic exchanges that drive this evolution.
And adding in the unresolved intricacies of the bat immune system, which has evolved to knock Bartonella down but not out, makes for a perfect reservoir to spread disease to less well-protected hosts.